As we start this topic, I want to acknowledge that initially we started this out doing these Zoom lectures to try to inform the public and also professionals that there is a significant alternative to toxic chemicals that are made by man that through a seriously flawed evaluation process are elevated to a term called pharmaceutical drugs, but they are still poisons, just as the father of pharmacology stated, Paracelsus, about 500 years ago, said, all drugs are poisons. The benefit depends on the dose. And to validate that, all pharma drug development starts out in different phases. And the first phase is toxicity studies. And you have to tell, for example, in the United States, the Food and Drug Administration, or FDA, how much of your substance that you want to make a drug kills half the research animals, and how much does it take to kill all the research animals? That should not be real comforting. But in our research, we had a major problem with the FDA. We could not kill anything. And so we couldn't give them an LD50 and LD100 with the active ingredient from the aloe plant. And we were thrown out of the drug approval process for Carrington Laboratories. However, in 1994, in the wisdom of our federal government, the Food and Drug Administration was involved with the passage of the legislation called the Dietary Supplement Health and Education Act, called DSHEA, which formally created the dietary supplement industry. Well, this, as we have gone into these topics, sometimes I have decided to address things that people are talking about, reading about, that really aren't involved with taking dietary supplements. And this is an example of one. And I want to address some of the problems about vaccinations that are much on people's mind because of all the problems that have been circulated in the media about the RNA vaccine for the coronavirus, or as I call it, the China virus, that has been a pandemic. So I will point out we've had politicalization of science, but that's not an old subject. There has been politicalization of science from the day one, and you will see some evidence of this in the history of the what is considered the most successful vaccine, smallpox, as I will share with you the development of this most esteemed vaccine. With that, I'll start with the first slide. I realize that the vast majority of lay people have the interest in dietary supplements or medications. What will it do for me? What benefit can I get? And there is no interest in the science, and they certainly don't want to be molecular biologists or geneticists or biochemists. What will it do for me? Fortunately or unfortunately, I like to try to spend some time to inform the public of why this can be a benefit to you. And vaccinations have really been in the public eye, and a lot of criticisms, particularly about the RNA vaccine. So we'll start with just what is a vaccine? Well, for decades, the Center for Disease for our federal government says it's a substance that prevents a disease. In other words, a procedure that you go through that's going to stop you from having a disease. Well, with the development of the RNA vaccine, this definition was changed, and the definition was changed to a substance that stimulates the immune system. Well, in stimulating the immune system, this RNA vaccine, and I hope we don't confuse you in getting into it, seems to confuse the immune system for some very important reasons that I will show you. Whether you interested with it or not, I don't know, but I would think you would like to have some knowledge of what you're hearing about to evaluate people giving pros and cons about vaccinations. The attempt to tell you that that a vaccine takes an infectious antigen. Usually it's a portion of the organism that causes the infection. And when it comes to viruses, particularly, there's a string of nucleic acids that are organized that gives the virus the capacity to approach a cell, enter a cell, pirate or take over the organelles within the nucleus that are an assembly line to make bioactive compounds that run the process of life. It's under the control of DNA, the genes. However, the infectious agent wants to take it over and produce viral particles. So if we were to say A, B, C, D, F, G were different portions of a virus, that you would inject this into the person, and then the immune system would make a antibody against the A, B, C, D, F, G. If it's not the self membrane signature of the person that's on all its cells. In other words, the infectious agent has in its surface primarily amino acids that make peptides or proteins that are different from the host that you're trying to protect. And this stimulation with this order of molecules on the surface of the infectious agent, there are certain white cells called plasma cells that produce antibodies against each of these proteins. And this antibody binds to the surface of any future encounter with the infectious agent surface molecules. And the titer sometimes goes down, but they're actually memory white cells that recall the activity of the vaccine that very rapidly starts making the antibodies to increase their titers to inactivate by binding to the surface of the infectious agent to produce what we call the vaccination effect. And I hope you can understand that. To show in a diagram, the yellow in this sling is an antibody that has a sort of Y shape. Actually, there's an electron micrograph to the right showing under the electron microscope that this is what an antibody looks like. And you have say a microbe or a bug, and it might be a virus or a bacteria, and we have little half round circles, each one of them representing, say, the A, B, C, D, or G, or antigens, are the proteins on the surface of it. Well, the antibody has a fixed end, which is solid, but at the tips of the Y, there is primarily written with carbohydrate molecules. It's kind of like a magnet that it produces a molecule there that will respond to the antigen from the microbe to bind to it so that it doesn't have its toxic effect in the body, and it keeps you from getting the disease. And we have over to the right a microbe too that has a sort of a different shape on its surface there. And you have to be specific for the antibodies against the antigens for the infectious agent. And so you have different vaccines for different infectious agents. And this binding neutralizes it by attaching this antibody to it to keep you from getting the infectious, and keeps it from being able to take over the cell and start making, for example, virus proteins and assembling them to make you sick. Now, I'm trying to get across to you the vaccine principle as it's stated, but one of the first things that I was surprised to learn, and maybe you do, but if you take 100 individuals, adults or children, and you give them a vaccine, not every one of them will develop a protective antibody titer against the infectious agent. And the stressing in this is that the site of entry of the infectious agent is important, because if you provide the vaccines at the point of the entry of this infectious agent, it's more effective. In other words, if it's a infectious agent in the GI tract, that if you gave your immunity through a fluid that you swallow, that would be better against that agent. If it's respiratory, through the nose and the oral pharynx and the down into the trachea and all, that would be much better than those. So the thing is very important. Now, our research with Texas A&M was very important in that they had a vaccine there for baby chickens, which to my surprise is the #1 vaccine that is present and used commercially worldwide, because chickens are put into houses with several thousand, and an infectious agent can spread and be very costly to have to either have the chickens die or have to sacrifice them because of their infectious. And this trivalent vaccine that they did research with at Texas A&M has the Marix virus, infectious bursal disease virus, and the Newcastle virus. And these tend to be spread through the nose. So the vaccine is a single drop of killed virus that is put in the nose, because that's the source that it's spread between the chickens, that it comes in through the nasopharynx. And so that's why this is an effective vaccine in chickens. And but and when you get into different types of vaccines, the portal of entry can vary. Like for the gastrointestinal tract is for polio, is they frequently have a little thing that goes through the mouth, it goes down, but then there's pulmonary, and of course there are infections that are spread by the sex glands through the urethra, the vagina, and frequently insects also like ticks and mosquitoes and other insects that puncture the skin, that the vaccine through the skin often is also involved with the vaccines against the sex organs, because the mucosa of the sex organs is similar, is a specialized skin. Now, we have been, I want to point out that the principle of the vaccines is not set and over, that it takes a while of stages of trial and failure to make anything work. And when you're flying in a Boeing jetliner at over 500 miles an hour at 35,000 feet, that is a long way from the first very short flying kite with a motor on it that the Wright brothers flew decades ago. That it's a lot of things happen, and in fact, a lot of planes crashed and a lot of people died before you got to the jetliner. And this type of experience to get safe air travel that we take now, that the development of vaccines go over a long period. And the RNA vaccine is a new way to try to develop a vaccine. And there are lots of questions that have come up that here you are giving a new type of vaccine to a child or adult that is perfectly healthy, and you want it to prevent the infection. But unfortunately, they found out that RNA shot does not prevent the infection like for decades what they were trying to do. And that's why they changed the definition to a substance that stimulates the immune system. And as I said, it's a dirty truth that everybody gets a vaccine doesn't get protection. And this is no different for the RNA vaccine, and we can hope that over the decades that it will be improved as they develop, much like airplanes have developed, that we will have a better vaccine because the theory of the RNA vaccine is an improvement over taking killed organisms or organisms that you put formalin with to or attenuated them until they run through cultures until they still have the signature on their surface, but they don't produce the disease. That's an attenuated organism that's used in sometimes vaccines. But it takes a period of development for vaccines, just like for airplanes. And we're right in the midst of this. But one of the things that I found out in vaccines was that there was a crash development of vaccines after World War II in the early stages of it, because the military was going into islands and continents where there were third world insects and all. And there was a white coat program developed of volunteers that they were primarily Adventist youth that would not carry a gun or serve in the military to kill or be killed, but they volunteered to be subjects for rapidly developed, particularly insects spread viral infections. And actually, some of these white coat volunteers died when they were tested against a rapidly developed vaccine, that it takes a while to do this. And this problem about vaccines is so loaded with controversy that in the United States, a major publication had to go all the way to Germany to find two molecular biologists to explain in an article why the RNA vaccine is a problem. And the reason I couldn't get a molecular biologist or a vaccine type physician in the United States is that politically would say it's good. Everybody ought to have it, but there were a lot of people that wouldn't because people were getting problems, which is not wanted for healthy people to be vaccinated, and then instead of preventing them from getting infections, many of them still got infections and they had problems from it. But the antigens on the surface are the molecules of the farmer vaccines stimulated antibody production. Well, actually, proteins in the process of the genetics of cellular synthesis, the DNA is the code of life that never leaves a nucleus. It's kind of like the reference books in a library. You can't take them out of the library, but you can make a photocopy of them and take it to wherever you want and work with it. Well, in the cell, RNA is made from DNA, which is a complementary copy. We'll go into the details what we mean by complementary, but the RNA dictates to the assembly line how to hook amino acids together to make the protein, which is the antibody against the infectious agent. And there are two steps that are important in this thing, is that when the RNA is made from the DNA, behold, in the genes, there is a audit step. Is the RNA correctly made from the DNA? And if it's not, it stops. And if you have the right molecules in your diet, it's repaired until it is the correct complementary copy of the DNA. Then the RNA is in two portions. One of a portion of RNA is the instructional RNA, and it goes to the assembly line inside the cell of how to hook all the nutrients together to make bioactive molecules to carry out the biochemistry of life. But a transport RNA is in the other portion of the RNA, which of all things goes out and looks for the amino acids, the fatty acids, the carbohydrate unit sugars, the vitamins, the elements, and brings them into the assembly line at the right time for it to be integrated into the assembly. So that's very significant. But there is an audit step for this interaction of the transport RNA and the instructional RNA, that when the protein is going to be made, or the sequence of amino acids, there's an audit step that says, is this genomic protein? And what that means is this protein that this RNA is going to give instructions, is it in the history of the maternal genes and the paternal genes of the mother or the father? Is it in their genetic tree? And if it's not a genomic protein, synthesis is stopped. And this is where a problem develops with RNA vaccines. the protein of the infectious agent is foreign. It is not in the genes of the host. And so what they add to this RNA vaccine are different substances to try to, shall we say, induce or fool the immune system to go ahead and make the antibodies. And this doesn't, at this point, work like the theory is, because why would somebody want to go to try the RNA? Well, the problem is with the actual organism that you try to either kill or make it attenuated, there is a certain percentage of individuals that will get the disease. And some of these diseases are very bad and even fatal. It's a low percentage, but if you could use RNA instead of the actual infectious agent, the idea is to make antibodies without ever being exposed to the agent that could give you the disease, to make the proper antibodies to bind to the agent. Well, this is not foolproof at this time, and I'm going to kind of give you the history of the most effective vaccine that's ever been done. But at this point, it seemed like the RNA at that step of where the second audit step is this genomic antibody or protein. Was it ever in the history of the male or the female? And if it isn't, it's stopped. Well, of course, it's foreign DNA, RNA, pardon me. And so they haven't figured out just how to get this to work just right. Now, why is this important? Well, that's what this slide is about. There are multiple systems in the body, and not only the immune system, but one of the major complications of the RNA vaccine is it's not generally known that the intrinsic blood coagulation system, and that's a system that if you're punctured or cut, that you don't want your blood just to continue to run out. There is a stepwise process of factors that clot the blood and keep it from going out. And one of the big factors in that is that it's in balance with a lysis system that's also in the blood, which Leiden factor 5 is very important. That as this coagulation process is able to go along, this Leiden factor 5 cuts the clotting material into little pieces, and it's called fibrin split products. All of us have a little of this, but this is a balancing system that you go and confuse the immune system. And this is why that one of the major complications of the RNA vaccine is strokes and blood coagulation, because it interferes with this balance of the tendency to have the blood where it's in balance with keeping it flowing, but also if you under conditions to clot and keep from losing your blood. Well, the RNA shot disturbs this balance, and strokes is one of the most common complications. And this is not the only system imbalance in the body that a confused immune system causes problems. Now, the World Health Organization declared that smallpox is 95% effective if you get skin pustules and a serum titer. And it's considered that smallpox, which was a major worldwide threat to life, liberty, and pursuit of happiness, that they even sent out words giving some thought about destroying all smallpox vaccine or all smallpox cultures, because it's no longer a world population threat because so many people have had the smallpox vaccination. Well, just like going from the Wright brothers airplane to a jet plane at 35,000 feet and 500 miles, let's go through the history of this smallpox vaccine to point out that vaccinations are not just a simple process. Is the smallpox vaccine as reliable as modern air travel? Did smallpox wipe out this disease as some people think? Well, in 1796, Edward Jenner, a physician, noted that milkmaids did not get this feared and lethal disease of smallpox. And so he took James Phillips, a little 8-year-old boy, scratched so-called cowpox lesion material on his arm. And though it was smallpox going around, he didn't get it. And now this has become a myth. And the question is why? Well, the vaccine comes from the term vaccia, cow, or vaccinia for cowpox, this story that I just got through telling you. But in 1798, Jenner published that this process was a lifelong protection. And as I told you, the CDC, you had this definition that vaccine is exposure to a material that prevents a disease, and that was what they claimed for the smallpox vaccine. But as there's politicalization in the 2020, 21, 22, there is, I'm going to show you that there has been scientific politicalization along the way for even the supposedly most beneficial vaccine there is. Well, in a individual named John Barron wrote the book, The Life of Edward Jenner, and he told for the first time that story of the milkmaids and this. However, there's a little problem. Cowpox does not make skin lesions. And so there has been a question about this story that this biographer put in this book that was written. And in letters that they can find that Jenner's wrote, he makes no mention of cowpox. However, there is a virus that causes horsepox, and they get crusted lesions. And there is the belief that probably Jenner took the cowpox and the horsepox, I mean, that made the skin lesion or the scar that's typical of a smallpox vaccination. And nobody knows exactly what happened then. And this Clarissa Dansico in 2018 examined as much as it could find of all the records, and that in 1813, 1817, some writings of Jenner actually stated that he used horsepox, lymph, and pustules from handlers of these lesions. And so you're still left with the story of the milkmaids, but they actually think that it was horsepox that it was done. Well, Dr. Charles Creighton, who lived from 1847 to 1927, was considered an unimpeachable accuracy source. And he wrote that there's no scientific proof that a particularly formal study was done that this scratching of the skin with this material that Jenner had prevented epidemics. And that he said that smallpox was not proven contagious or cause epidemics. And that's a very complex thing to actually go through the process to prove that an organism causes a disease. So he said that there was no proof that it worked, that it was safe, because no scientific studies were done. They just started doing it. And there were some prominent physicians at the time, Wilson, Hills, Martin, Gardner, and others that testified publicly that this procedure by Jenner caused disseminated leprosy, which was one of the poxes that go all back to biblical times. In other words, they said, here you claiming it stops smallpox, and these physicians says it causes leprosy. That also Robert Blackwell, a noted international leprosy physician, said he took samples of Jenner's vaccination and could produce leprosy. In 1799, Dr. Drake vaccinated 3 children, and when challenged with the smallpox, all 3 developed smallpox. Dr. Creighton publicizes failure in the newspapers and kept telling of multiple failures of this vaccine up until 1804. Here is a series of individuals that from history that the House of Medical Supplies in England said that they had 56 cases where Jenner's procedure had been done and they developed smallpox. That Lord Petty of the House of Commons said that Dr. Jenner was a blind, conceited person, that his vaccines had nothing but failures, and he gave multiple examines. And then 1810, in a Medical Observer magazine from England, they had 535 cases of smallpox after vaccination. And 97 of them were lethal. And 150 of them were scarred for life by this procedure. That at this time, 10 medical professionals supported these claims that were in the Medical Observer published. In the Medical Repository Monthly Report, they reported that multiple people were vaccinated, get smallpox, and that many died, and that Jenner's vaccine was of no value. A Scottish physician claimed that he had 1,200 cases of Jenner's vaccination, that many of them developed smallpox after this vaccination, and some died, and that there were confluent lesions all over the body that complicated these diseases. In a journal book by Colbert, written called Advice to Young Men and Women, he wrote that many people get smallpox and nearly die from a lethal disease, and that the vaccination does not protect them. That's not the end of the criticism. As I say, there's been political opinions in science, just like in politics, in government issues, that Humphreys investigated the United States history of the Jenner vaccine. And he said there's no decline in incidence of death despite a high percentage of vaccination with this procedure. The Hartman developed a organization of physicians against vaccination because he said that in the United States they had the same results that they had in Europe, that the vaccinations did not prevent this disease. That in England, in France, and in Prussia, they had large numbers of individuals that in this group they had vaccinations, and yet they had thousands of deaths and thousands of cases. And in Germany, they claimed that in a group that 96% were vaccinated, they had 30,742 cases of this disease, and that 1 million died. That Italy, they had epidemics of the smallpox with 18,110 deaths. In Japan, a huge number of vaccinations and deaths. So this was what's going on in this history. That in 1844, they had a smallpox epidemic with 647 admissions of people that had had the vaccination. They had a series of 312 that vaccinated, that 48% of them got the smallpox, and 24 died, or 8%. That 100 had a mild case, and 2/3 had a moderate to severe case. In 1845, in Lancet, they stated that the Jenner vaccine for smallpox was not as effective as expected. That they had, despite high vaccination rates, they had epidemics in Boston in the years listed there. And then in Chicago, with 90%, 5% vaccination, over 2,000 cases, they claimed 500 died. In England in 1857, there was 14,244 died. And they had these other epidemics in the number that died. And that in Lancet, a long accepted published medical journal, they claimed that 17.5% of people that had been vaccinated died. However, in England in about 1900, they stopped having smallpox. And the big question has always been, why did they stop having the effect? And this is a graph of the U.S. of smallpox cases since 1949, that since 49, there have been no epidemics in the United States of the smallpox. And these peaks there are before 1949 of different epidemics across the United States, particularly in Boston, that the question arises, why in Europe and why in the United States, after these dates, there were no epidemics of smallpox? And as I said before, after 1900, in the United Kingdom, England, and Wales, each one of those peaks are an epidemic, but no smallpox after 1900. And this is why the World Health Organization has reported that if you get pustules in your skin and a little scar, and particularly if you have serum antibody levels, that there's a 95% protection. And even today, no formal vaccination studies have been conducted to take a group that is randomly selected and vaccinate them, and take another group and vaccinate them, and then challenge them with the smallpox virus. But in 1966, 33 countries outside of United Kingdom and the United States did have smallpox epidemics. And so the World Health Organization launched a eradication program to try to go into these 33 countries and give them the vaccine. Yet the questions were being asked, why were there no cases after 1900 in Europe and 1949 in the United States? And nobody's been able to answer that question. The theory is that in the genetics of a group of humans, that you can acquire an immune resistance to a infectious disease. And this is a natural adaptive immunity. And that again, we pointed out earlier that there are 5 barriers to human innate immunity, that how you administer the vaccine is very important, but it should be to be effective, so that when the body's immune system is made up of the barriers to infections, just like the skin, the mucosa of the nose and the hairs in the nose and the lasted eyes, and actually tears have some anti-infectious properties. That the epithelial cells excrete molecules that are interferons that will bind to viruses and inactivate them, and that there are innate immune cells, dendritic cells that are shown there, and white cells and macrophages, and that in the white cell population, there are T cells that come from the thymus that make cytokines and interferons and interleukins, which are humeral molecules in the serum that circulates in the blood that are anti-infectious. And if, of course, if the vaccine is able to activate these innate immune things, that we get down to the beta cells, which are other types of white cells that make antibodies. But emphasize again that these different cells have to be activated to produce their resistance against infection. And yet, since the dates that we showed you, they haven't done it. Now, an unexpected research that got doing is they found out that you can give vaccinations against measles, but the antibodies are not required in measles to cause resistance to getting the thing. And it's these humeral things. In history of immunity, there was a major conflict between Germany and France, Bering in Germany, who said that host defenses are in the serum. And of course, this is where antibodies, you can't see them, they're tiny molecules, and acute reactive phase proteins and toll proteins are present that are anti-infectious. And there are the measles, you don't develop antibodies. It's these humoral factors that are made by these cells of the immune system that circulate in the serum that protect you against the virus, and they do not make antibodies, which is a truth that causes questions about the efforts to try to make effective antibodies. But for gold standard for decades, we recognize that the bubble boy phenomenon, that they cannot make antibodies, and they 1960 they had a bubble boy that recovered from measles because those children can't make antibodies, and that was amazing because they thought that the measles would probably kill this child, but this is how they first began to do research and found out that the vaccine against measles doesn't produce antibodies, because here was a bubble boy that doesn't make antibodies. And at this point, I might add that one of the amazing events of the decade that I was involved with the international dietary supplement company was that a bubble boy up in Massachusetts began to make antibodies, and they didn't have to give gamma globulin shots to him by adding the aloe oligosaccharide, which is in New Eden, to the diet. And this so-called bubble boy was able to come out of the bubble, and he didn't get infections because of giving more comprehensive nutrition built around the molecule that is in the aloe vera plant that boosts the body's host defenses. And so the point we were making with these slides, there are multiple factors in host defenses against effective agents. And of course, with a vaccine, you're trying to activate all these defenses. So what we end up saying is that the principle of vaccines is not fixed or final. There are lots of questions about how individuals can get acquired defense against an infectious agent, just like how terrible smallpox was, and how ineffective the vaccine was, proven over and over again. And yet after a certain date, they have no more cases of smallpox in England, and no more cases in the United States, though when they were giving extensive vaccines, people were getting it. And one of the other things that they point out is that in society, we have developed refrigeration and sanitation procedures from the slaughterhouse all the way to the market, where you're buying in packages food, that the refrigeration improved hygiene, wearing masks and gloves, and preventing foods from being inoculated with infectious agents can reduce the incidence of infections. That we stress that given the vaccine, that the portal of entry of this different infectious agents seems to give more benefit to the effectiveness of the vaccine. And there's no question that the RNA theory of the RNA dictates to the assembly line how to make an antibody without ever being exposed to the infectious agent would be an improvement, and we could eliminate this small number of individuals that get the disease. But it has not been perfected. And I want to say that the theory is good, but it hasn't been worked out any more than when the first airplanes were flown and crashed and many people died trying to develop it. There is much more work that has to be done before the RNA theory that the coronavirus is built around is perfected. And if it can be perfected, it could be a major improvement in vaccinations. And those people that are involved in this research are very hostile to criticism about the RNA vaccine. However, there's no question that there are problems and they haven't been solved. But I hope that you have a little better idea about what the intent of vaccination is. And hopefully, I would like to see the CDC eventually alter its definition of vaccine back to a procedure that exposes the individual to a method that prevents them from getting the disease rather than the stage that we're in right now with the RNA vaccine, which is far from being perfected. I hope this is worthwhile discussion about vaccines, and you have a better idea of what the medical profession is trying to do to prevent the terrible problems of some of your infectious diseases that can even cause death. So with that, that's the end of this lecture about vaccines, which is so much in the media that surrounds us. And we've got these voices saying that one thing and others that say just the opposite. And the final answer is not present.